Okay, hello everybody. Welcome back to almost an episode 13. Lecture 13. Definitely done more quote unquote episodes than 13 this so far. But yeah, welcome back to lecture 13. Or welcome to lecture 13. Gonna cover enzyme mechs. This is a bit of an interesting uh is an interesting lecture because they kind of just go through enzyme mechanisms, which I don't know if you have to have these memorized. I personally didn't have to have anything memorized. We did have to understand basically how the serine protease worked, with a base ripping off the H and making the nucleophile for the serine. But other than that, we didn't really have to know much. I think they're doing the same here. You might want to keep some like ideas in mind for how these work, but I'll basically cover these and try and explain them to you guys so it makes a little bit more sense if it was confusing. So looking at chymotrypsin and enolase looks like. Um, yeah, let's get into it. Crazy slide. Cool. I don't really think you need to worry about this. Enzyme mechanisms need a lot of things. Read through this if you want, but I feel like based off what you've learned and we've covered in the lectures during the reviews and what you've done in lecture, like actual lecture, you should have a good idea of how this works, right? So here's an example of enolase, right? Converts phosphoenol pyruvate into pyruvate, penultimate, right? The step before the last, right? That's all penultimate means, right? Um, so yeah, we go from phosphoenol pyruvate to pyruvate, right? Um, but yeah, it's actually technically, I guess technically it's the final step of glycolysis, if I'm not mistaken. But anyways, um, we've got a couple things here. So we've got a metal ion to help stabilize, right? So this also lowers the pKa of CH by inductive effect, right? And a local environment, yep. So it impacts the pKrs for lysine and glutamate with the actually, yeah. So a whole bunch of stuff here, right? Basically, what they're trying to get you to understand is that these magnesiums, they'll basically pull on the electron density of and basically stabilize, right, the electron density of these oxygens. So these would affect Km, right? When you think about that, right? Maybe they would affect calic activity as well, right? But for sure they help with binding affinity, right? So and that allows this lysine to act as a base, right? To grab this H, right? And then that allows for the electrons to go down, become a double bond here, and you move on to this step, right? So now you're here, right? These electrons on this oxygen come back. That causes this to flip over here, and that causes this bond to break off here to form a water molecule that ends up leaving, and you end up with phosphoenolpyruvate, right? Hopefully that makes sense. Basically, these enzyme mechanisms are just a bunch of OChem. So if you remember OChem mechs, then this should be very easy for you. So digestive enzyme, serine protease, yeah, so here's it, right. So um catalyze, yeah, so carboxyl groups. So it cuts the carboxyl side of aromatic amino acids, right? For chymotrypsin, right? How does it know that? Well, before we even think about that, going into the mechanism, let's think about how it would be able to identify phenylalanine, tryptophan, and tyrosine. What do they all have in common? They all have these hydrophobic aromatic rings. So I'm expecting the binding site to have a lot of hydrophobics so that there can be some like interactions, right? So maybe some valines, isoleucines, leucines, maybe some more phenylalanines, tryptophans, and tyrosines, right, in there, so that they can attract these phenylalanine, tryptophan, and tyrosines in the actual protein that they're trying to cut, right? And so that allows the stabilization of the enzyme substrate complex, right? So I'm expecting a pocket that has hydrophobic residues in it, right? Because I need the enzyme to be specific for these residues, right? They need to be oriented in such a way that these rings are the only things that can bind. So things like just just a single valine or a single leucine or isoleucine are too small to fit in that pocket. It has to be a big ring like these three. Right, so serine protease. Protease, by the way, if you don't know, is just something that cuts up proteins. Right, ACE is just enzyme, right? Protease is just a thing that cuts up proteins. Right? Um, and yeah, so why is it biologically important for the rate to be very high? Remember that your body needs these. So basically, it's so slow if you don't catalyze this, right? And your body needs these proteins and amino acids to make its own proteins and stuff like that, right? And these are helpful for digestion, right? So if you eat food, right, there's a bunch of proteins in your food. If you break down the proteins, you can use the amino acids for other things in your body, right? If you don't have enzymes, you'll eat food and you'll spend so long trying to break down these proteins, and you'll probably just be dead, right? Because you can't make any proteins, and your body's just stuck doing nothing. So it's important to have this rate enhancement with chymotrypsin as the enzyme, because it allows for quick breakdown of these proteins that can be used for proteins that you actually need, right, when you eat food. Structure of chymotrypsin, great. I don't think you have to know this, but just keep. So remember we talked about hydrophobic pocket, right? So the aromatic side chains can fit in it. We already predicted this, right? We knew this was going to happen, right? There needs to be some sort of hydrophobic pocket. And what amino acids need to be in there? Leucines. I almost said arginine, not arginine. Leucine, valine, isoleucine, phenylalanine, tyrosine, tryptophan. Those kinds of amino acids, right? And yeah, here's a crazy complex image of how this works. We'll do this step by step, right? So here's the free enzyme, right? So we can look at a bunch of these, right? So notice how we have this aspartate here pulling on this H, right? Which makes this more basic, right? This histidine is now more basic, right? Probably has a pKr higher than 6.1 if I had to guess, right? And so it's now forming this not hydrogen, like this hydrogen bond, right, with this serine here, right? We've got a glycine here, and we've got our hydrophobic pocket. Lo and behold, right? We would have seen that coming, right? We talked about this already, right? Has to be a hydrophobic pocket. And then we have our oxyanion hole, which is basically where our substrate is going to come in and not bind, but basically sit. This is where the substrate will actually bind, right? It'll sit, right? And it'll allow us to do our actual work. So here's our substrate. We've got a phenylalanine here and the two other substrates, two other amino acids on the side, right? You can see here the mechanism at work, right? So the hydrophobic pocket binds this phenylalanine, right? It's large enough, and the residues around here are very specific for these aromatic rings, right? And what happens is, once we have bound the substrate in here, this histidine is now basic enough that it removes the H from this serine, and this serine now attacks this carbonyl and makes this oxyanion, right? So this basically these electrons come up here, right, and we form this negative oxygen species, right? And now what you should know is we are in a covalent intermediate where the serine is now bonded to the substrate, right? And catalytic triad, by the way, just means which enzymes actually participate in the actual reaction. Okay, so now we have this covalent intermediate here, right? All the change. So notice, right? The only change that has happened here, right, is that this serine, right, this serine right here, that's initially here, has now formed a bond here, right? And see, look at how once you've done that, right? You see this negative charge here. This glycine and this serine's N terminus, right? The N terminals of the serine, right? It's not technically not the N terminus, but the nitrogen of the serine amino acid, right? And the nitrogen of the glycine are now forming hydrogen bonds with this oxygen, this negative oxygen, which basically stabilizes it, right? Keeps it stable and doesn't cause it to cause some sort of massive explosion or whatever. It's obviously not going to explode, but basically the delta G of this state is is relatively low in comparison to what it would be if you were to do this without an enzyme, right? Because there's no stabilization without the enzyme, right? And so now, we're back here, and we can now take the electrons from this negative oxygen, put them back down, and we can kick off this big group here. This is our leaving group, which is this the remainder of the peptide chain, right? And this is just like an esterification, right? Where you form this tetrahedral intermediate and you kick off one of the groups, right? It's just an esterification reaction from 239. right? And so now you're left with this product, right? And all we've done to go from here to here, right, is we just brought these electrons down and kicked off this group here, right? This group's been kicked off. These electrons have been brought down, right? And now you're here, right? And this is our leaving group, right? And now you're done with the reaction, right? So now, right, you basically go through the same reaction again to replenish the catalytic triad of this protein, because you need the protein to be usable again, right, after it's done. So how do we do that? Well, let's think about what has to happen, right? So what has to happen is, right, this massive protein group needs to leave, right, and this serine has to get its H back. So basically, this serine has to leave, right? Well, how do we do that, right? Well, we just add water, right? So if we add water, right, what does the water do? The water forms a hydrogen bond with this nitrogen, right? Where eventually this nitrogen deprotonates this H, right? And this OH minus group now attacks this carbon, right? And we're doing that tetrahedral intermediate again, right? Another transesterification, right? These electrons kick up, and then you should expect what happens next, right? You form this oxyanion intermediate, right? And then eventually these electrons come back down, right? These electrons from the bond, right, from the serine, right, get kicked off to grab the H, right? And now you're back here, right? Notice how that happened, right? I just want to make sure I get something right here. So, because it looks like they do sometimes make these a little bit confusing to read. But so this OH group, right? Hold on. Yeah, so yeah, this is a bit weird. So basically what they've done, right, is these electrons have kicked up, right, onto this oxygen here, right? And they haven't done a great job of explaining how this has happened. But basically, right, this bond here attaches this water molecule, or the former water molecule, because remember we actually removed this H. It actually attaches it to the carbon, right? So the water is actually this group right here now, right? That has been deprotonated, right? So it's been deprotonated, right? And these electrons have been used to actually attack this O minus, right? So the tetrahedral intermediate, right? And this is a serine, obviously, right? Right, this group here, serine. So, right, now you just need to get rid of the serine bond. So these electrons from that are bonding to the protein, right, or a peptide chain, will now grab this H that's now attached to this histidine, right, that we initially grabbed from the water molecule, right? And now we've replenished that catalytic triad, right? Now we're back to where we started, right? And then this group obviously leaves, right? And we're back to where we started in the first place, right? So if we go here, you got mechanism-based inhibitors, and you got to think about what these inhibitors do, right? So you're like, okay, well, in order for this mechanism to take place, right, we need the substrate to bind in this hydrophobic pocket. But what if we bind something else there so strong that it actually doesn't want to leave, right? That would stop the catalytic activity of the enzyme, right? And lo and behold, here's an example of that. This binds so strongly into this pocket, right? Into this pocket here, right? That no other substrate is able to come in and bind to that area, and so the protease is just inhibited, right? So this would probably be an example of a competitive inhibitor, right? Because it's structurally similar to the substrate, right? So, right, so again here, so often but not always looks like substrate, right? This is probably competitive, not too sure, but probably competitive because it looks similar, right? It binds in the active side of the enzyme, right? Forms a, well, oh, actually, sorry, in this case, it actually forms a covalent complex and is very dangerous, right? So it's irreversible in this case. So what probably happens, right, is that this sulfur here is actually quite electrophilic because there's a fluorine pulling here, there's an oxygen pulling here, and there's an oxygen pulling here. So what probably ends up happening, right, is this what, are they showing it here? Yeah, they're showing it here, right? Is that the serine, I was going to say, the serine, right, actually attacks here and kicks off the fluorine, right? And it gets stuck in that form, right? So it gets stuck in this form, right? So the fluorine has left, right? Right? And so on top of that, right, this phenylalanine group most likely sits in this hydrophobic pocket, right? And further stabilizes that inhibited state, right? Oh yeah, this is fun. So this is actually how penicillin works, if I'm not mistaken. Um, but peptidoglycan is a peptide plus sugar, right? Peptide, peptido, protein, glycan is sugar, right? It's a protein sugar complex basically that is the major component of bacterial cell walls, right? Um, and they use transpeptidases, so these enzymes, right, that form these bridges between layers to cross-link these walls, right? Penicillin. Well, when we look at that, almost like I predicted this. I actually didn't even look at this beforehand. Um, so yeast produce penicillin, right, which attacks transpeptidase to inactivate it. This is penicillin, right, or at least a part of penicillin, right? And the cool thing about this is the enzyme, right, transpeptidase that's used to build these walls has a serine on it, right? But if you remember from OChem, these four-membered rings are so reactive, it's insane, right? It's so unstable. There's a little bit of resonance stabilization here between the nitrogen and the carbonyl, right? But like these are so unstable, generally speaking, right? So it's not unstable enough that it just explodes, but if you're given an opportunity to react it with something, it'll definitely react, right? And the transpeptidase enzyme that's used to actually build these cell walls or these peptidoglycan walls in the bacteria actually takes advantage of this and actually gets inhibited by it. So it'll attack this carbonyl, right, to break this ring, right? And you end up with this product, which is very, very stable and actually ends up killing the bacteria because the bacteria can't produce its walls anymore. That is how penicillin actually works. Um, but yeah, so the thing is though that eventually the bacteria figures out how to stop the drug by mutating enzymes, basically through evolution, right? Um, and natural selection, right? Because eventually you'll get some mutations, and those mutations will stop the drug from working, and then you have to create a new version of the drug to actually get the thing to work again, right? So in this case, right, got penicillin, right? The reaction takes place like normal, right? But now the um when the water eventually, right, at some point, right, water will come in, right, and it'll basically kick off this group, right? And this version of penicillin does not work anymore, right? Because it's been the cyclobutane has been destroyed, right? And that cyclobutane is what allows for the reaction to occur, right? So you have to make another version of it, right? And so in this case, you make another version, clavulanic acid in this case, right? And so, right, so you do this, right? And this version is a little bit different, right? But it's the same idea, right? It's just that these uh these slight mutations in the enzyme from the bacteria force us to have to change bacteria, change drugs a little bit, to actually get things to work. So notice how this extra nucleophile has been added on, probably due to a mutation, right? But in order to form a new drug that actually binds very strongly, we actually need to make a section here that has an electrophilic center, right? So that this nucleophile can attack, so we can inhibit the enzyme. Okay, cool. Definitely be sure to, you should be able to interpret catalytic steps, right? In terms of catalytic strategies. So just be aware and try to understand how these mechanisms work. What amino acids would I expect for certain things? So if I wanted to protonate something, if I want something to act as a nucleophile, if I want something to act as a base, if I need a hydrophobic pocket to fit something in, right? What kind of amino acids do I need for all of those, right? You should be able to tell, if I mutate something or if I change the pH, right, how does that affect the catalytic activity of this enzyme? So for example, like if I need a lysine or an arginine to act as a base, right, for something, if I lower the pH, right, and protonate them, they're probably not going to be as basic anymore, right? So you can imagine I probably destroy the catalytic activity if I do that, right? Things like that, right? If I need an aspartate to be, if I need it to be a proton donor, right? Maybe if I increase the pH too much, maybe it just will be deprotonated automatically and it won't be able to do that job anymore, right? So change the pH, change the enzyme, mutate it, see how that affects the enzyme. Think about what that does, right? Make up an enzyme. Think about what that would do, right? And just keep in mind, what are they? How do they work? With Michaelis-Menten kinetics, right? Remember Michaelis-Menten kinetics, right? How does that connect to all of this, right? How does Kcat and Km connect to all of this, right? And this why are they useful in medicine part is really cool because your body is made of enzymes, right? These enzymes are what keep you alive, and it's just really interesting to think about. But yeah, I guess if you want to look at these books, go for it. But yeah, that's the end of this one. I hope it was helpful. Definitely on the shorter side, but definitely not. This lecture I wouldn't say it's the most important lecture of them, but definitely helpful because they will ask you mechanism questions most likely. Not like arrow push mechanisms, but they probably will ask you questions about what if you mutate this? How does that affect the activity or Km or Kcat of the enzyme? So, but yeah, hope this was helpful. If it was, great, and if it wasn't, yikes. If you have questions, feel free to come by the IC. More than willing to help. But yeah, have a good one, guys.